Thanksgiving week, and welcome to our Viruses and Vaccines seminar. Today we have Dr. Emily Martin from the University of Michigan, who will be presenting to us on influenza epidemiology and vaccine effectiveness studies. So Emily is associate professor in the Department of Epidemiology at the University of Michigan. She completed her PhD here at the University of Washington prior to joining faculty first at Wayne State and then the University of Michigan. She serves as the co-director of the Michigan Center for Respiratory Virus Research and Response, has really led a lot of the important work around understanding household transmission of respiratory viruses, particularly influenza, and is part of the very impressive University of Michigan research program on viruses. So thank you, Emily, for joining us. And then prior to Emily's talk, we'll have Trevor give us his update on SARS-CoV-2 genomics. And for our next seminar, which is on December 19th, we'll have Addie Stern, who is associate professor from Tel Aviv University, and will be speaking to us on molecular genomics. So I'm going to go ahead and stop sharing and go ahead and turn it over to Trevor. And as a reminder, if you could put your comments in the chat, and then we'll address them either during the talk or at the end.
Wonderful. Thank you so much, Helen. I'll try to keep this brief. I think Pavitra went over some of this last time around. So here we go. So I think we all are basically aware of this, that there was an early wave that peaked in September in the US and many other places. The difficulty, of course, being that cases are no longer basically reported, so it's really hard to understand what's happening in circulation patterns. The main data that I've been looking at here is wastewater surveillance, which shows you see that the recent peak is not what it was, say, in summer 2022 with BA5 or the BQ1 XBB wave last winter, but it's still significant compared to this summer. And then we also see this in hospitalizations as well. So again, this is kind of dampened compared to what we'd expect for true circulation, but it does give us an idea of actually what's going on. So we have kind of a trough in the summertime of daily hospitalizations that peaks in the US in September-ish, and it's slowly coming down, but not coming down that quickly. And then the main point here is that we see much of this increase from here to here is really driven by the takeoff of EG5.1. This is a derived XBB that is basically the current vaccine strain that hopefully everyone here got, that has the additional EG5.1 has the additional spike mutations 452H and 456L. And 456L is really kind of what helped to propel it to increase in frequency. And we can see this remarkable pattern that kind of basically keeps occurring, where if we look at, say, site 456, that in the last year-ish, since maybe spring, we've had a bunch of parallel evolution. So multiple lineages, multiple clades of SARS-CoV-2 evolved this 456L mutation, and that mutation is fit. It's good on all of these different backgrounds. So we see it takes off again and again and again and again. And it basically just happens that EG5.1 was like a good background. And so with other mutations there, that's the one that outcompetes with maybe a bit of circumstances as well. And this pattern we see is probably partly due to epistatic interactions, but I think mostly due to actually changing population immunity. And you imagine as like people develop antibodies to the old version, then that creates kind of opportunity for, yes, now 456L is a really good escape mutation. And so it spreads in these different backgrounds. And then really interestingly, we see on top of 456L, 455F. This is now the latest mutation that's behaving like this. So we see kind of again and again, yellow to blue, where 455F is taking off and taking over. And what's also really interesting here in this kind of epistatic, actual kind of molecular epistatic interaction has been worked out, that 455F is not good by itself. It needs 456L to be good, to be fit. And so that's why we only see kind of green to yellow to blue rather than just F without L. And so now we're seeing that this 455F is spreading rapidly. This is our best guess at global frequency, where it's gone from 1% 5 months ago to 60% of global SARS in just 5 months. And this rate of spread is extremely rapid, much faster than flu, and this kind of process is then ongoing. Much of this is in China, where it's predominant, but it's now spreading throughout the world as well on these different backgrounds. And then finally, the other major story that's going on is, as I think most people are aware, there's this BA.2.86 variant that emerged as a saltational variant, very similar to Omicron, also having 30 spike mutations. We're just not calling it a VOC. It was first detected in August, probably emerged in May-ish in Southern Africa. The initial kind of outgrouping isolates are from Southern Africa, and then spreading to the rest of the world. Despite having all of these mutations, it wasn't actually that much better in terms of escaping neutralization than XBB.1.5, et cetera. So it stayed at basically low frequency. So if we look at places where it's detected well, we see kind of it's been increasing slowly, say in the United Kingdom from September to November, but for the most part, decently slowly. I realize I don't have the US in this picture. However, what's just happened is, of course, we have 455F that appears to be good in all of these different backgrounds. That's now occurred on top of BA.2.86. And so that sublineage of BA.2.86 that has spike mutation 455F is called JN1. And so if we look at JN1, we see that it's taking off much more rapidly across places in Europe primarily. And at the moment of kind of everything that's circulating, when we do these trackers to figure out frequency change, we see that JN1 has the strongest growth advantage relative to the background strain of XBB.1.5. And so if we kind of pause mutations at this point, we would expect JN1 to take over the world in the following months. And here's BA.2.86, so it's a big jump having 455F on this particular background. And so yeah, we'll see if this occurs or if something else kind of occurs to compete with it. But this is kind of where we're headed, at least at the moment. I'm going to stop there. Stop sharing. Yeah, and please just ask any questions in chat. I'm really looking forward to your talk, Emily. Thank you.
Thank you, Trevor. Emily, do you want to go ahead and pull up your slides?
Yeah. I have so many questions about JN1, but I will wait until later. Okay. All right, I'm assuming you are going to stop me if this does not show up properly.
That looks good.
All right. Excellent. So what I wanted to talk about today was talk about some of the issues that we're seeing in influenza epidemiology, and particularly issues that are affecting vaccine effectiveness, both in actual effectiveness, but then also issues that are affecting our ability to measure vaccine effectiveness with the speed that we need to be able to inform vaccine policy and public health policy, and how that's been exacerbated by the emergence of SARS-CoV-2. So the theme, the conclusion of this talk, I'm going to give you the conclusion first. The conclusion of this talk is it's tough to make predictions, especially about the future. And I love showing, this is a picture from the CDC photo bank. And I love showing this picture to my students because, and when I tell my students that this person is looking at this plate to figure out what vaccine strain we should pick for next year's flu vaccine, they don't believe me, that they don't believe that that can possibly be true. And then when I say, and then even better, those red blood cells in that plate, they're not human red blood cells. They're probably from turkeys or guinea pigs. And then they really don't believe me. And so this talk is really going to be about why is it so tough to make predictions about the future when we're measuring vaccine effectiveness and then trying to apply that information going forward. In the fall, this time of year, I think anybody working in virology is used to getting phone calls from people in the media who say how effective is the flu shot this year. And I really liked this article from Fortune. They got it right. Where how effective is the 2018 flu shot? They say the short answer, no one really knows, and we don't know how effective these vaccines are until we start to see. We can try to make predictions based on the sequences, but we really don't know until we start to see what the data looks like on the ground. So this is probably this is probably known to this crowd, but I wanted to walk through the yearly vaccine selection process because it really does impact the significance of the different results I'm going to talk about. So we use global surveillance data. And now we use a lot of sequencing and then HAI work to inform strain selection that's done by the World Health Organization. It's then ratified by the US, by the VRBPAC at the FDA. Then seed strains are, for most products, still grown in eggs. We've been tracking different product uptake in Michigan, and eggs still is the predominant product all around the state. And then the eggs grow very slowly, and eventually the vaccine is ready, and we inject it into the arms of happy children everywhere. And then that's where my job starts. We then do the surveillance both to see what viruses continue to circulate throughout the subsequent flu season and what the vaccine looks like in that context. I'm going to show you data from a number of different programs that we have going on here. It's going to be a combination of data from our CDC and NIH partnerships on the right in the respiratory virus research and response group here at Michigan, from hospital-based vaccine effectiveness surveillance, ambulatory-based surveillance, and some longitudinal programs looking at community households. And then on the left, I'm also going to show you some outbreak response data from our partnership with the Michigan Department of Health and Human Services. And it's through that partnership that we are part of CDC's Insight Net now as well, where our role is to be working in tandem with the health department to support their response efforts with analytics. And I'll show you a little bit more about what that looks like. So I've already said vaccine effectiveness probably 20 times. I'm going to say it 100 times more in this presentation. And I am frequently surprised by how often people don't realize what vaccine effectiveness means. And so I wanted to show this quick graphic so that we're all on the same page. Vaccine effectiveness is the percent reduction in the amount of illness you would have experienced in a community if the vaccine had not worked at all or had not been delivered. So here on the left, we have a community that has entirely received a vaccine with 95% effectiveness. Normally, all of these individuals would have gotten sick, but we only have these green individuals in the bottom right corner that have gotten sick with a 95% effective vaccine. If you go all the way to the right, you have 60% effectiveness, which means 60% are now these blue people that had illness averted, and then the green people are people that still got sick despite getting vaccinated. So the reason I bring this up is because this is our typical flu vaccine effectiveness running up to just prior to emergence of SARS-CoV-2. These numbers oscillate between 40 and 60%. And then in some years when the vaccine is really off, we're seeing numbers like 10% or 19%. It seems this, our brains, if a number is below 50%, our brains will often go to this thought of, it's a coin flip, right? The vaccine isn't even as good as 50-50, right? That's not what that means. That means that this year in 2016, 2017, I'm going to show you some data from those seasons specifically. It means it was a 40% effective VE estimate. It means that we were reducing the burden of ambulatory infections by 40% nationwide. And flu is so common, and medical visits for flu are so common that it actually is a very substantial absolute number of illnesses that have been averted. So I'm going to start before the pandemic and then kind of move us forward in time. And I'm going to start with 2017, 2018, and the case of the egg adaptation. Show you some of the work that we've done around egg adaptation of viruses during the vaccine production process and what impact that has on vaccine effectiveness. So this is data coming from the Haven studies. This is a CDC-funded network in Michigan, Pennsylvania, Tennessee, and Texas. We did a pilot year of this in 2014, 2015, and then there was the national network through 2020. This is a test negative design study where you look at the vaccine effectiveness and prevention of hospitalization due to influenza. In that first pilot year in Michigan, actually, we found that the vaccine effectiveness and prevention of hospitalization, those numbers, the point estimates were actually higher than what we were seeing for vaccination in prevention of mild illness. So it was preventing severe illness more robustly than it was preventing milder illness, which at the time was a newer idea and a more kind of, we were surprised, we didn't know what to do with it. Now in the COVID era, we're very familiar with that pattern in VE estimates. And as part of the pilot year, another thing that we did was we collected the residual specimen from people as they were being admitted to the hospital, their earliest blood specimen. And then we tested those blood specimens against HAI and NAI for antibodies against the vaccine strain and the circulating strain. And what we found is you can take that acute blood, and within that first 6 days of onset, the HAI titer we didn't see any population patterning in the HAI titer compared to people who were admitted later versus people who were admitted sooner during the course of illness. And so we started to use these bloods as a way to look at and try to approximate what the pre-infection antibody titer was in people coming to the hospital for an acute respiratory illness. And here for HAI, we saw that as the titer grew along the x-axis here, with the higher titers, the percent infection went down. So we actually did see evidence as well that what we were measuring mapped reasonably well to what we would expect from a correlate of protection. So 2017, 2018 was a year in which we had a predominantly H3N2 year, although there was some B Yamagata here, but most of the sequences were coming back H3N2. And the H3N2 that was being identified was from the subclade 3C2A. The Hong Kong, the 2017-2018 vaccine strain was a Hong Kong 4801-2014 vaccine strain, and this was also purportedly 3C2A. And so we should have expected to have a match between the vaccine and the strain that was circulating. Side note, I didn't make a slide specifically about this, but I love seeing this much B Yamagata here, since for those of you that don't know, since the emergence of SARS-CoV-2, we actually have not seen B Yamagata come back at all. So while we're struggling with understanding influenza since the beginning of the pandemic, it's good news that we may have accidentally eradicated one virus. So that's a 1 point in the win column for us. So this is the data looking at the adjusted vaccine effectiveness preventing outpatient medically attended influenza illness. So this is the nationwide data in 2017 and 2018, and H3N2 is right here. And this, so we're seeing a range of, depending on the age group you look at, most of these estimates are 20% or sub 20%. This does not look like the data that you get from a vaccine that's well matched. This looks like the data that is a mismatch, but the broader clade number was similar between what was in the vaccine and what appeared to be circulating. And 3C 2A during the 17-18 season. So these are all, here we've got sequences from individuals that were coming to the hospital as part of Haven. So here are the sequences. With anything that starts with MH is somebody that came to our Michigan site in Haven. These were all sequenced by Adam Loring's laboratory. And you can see that we have a bunch of these sequences in what is 3C2A2. Here we have the Hong Kong vaccine here in the middle. And so we've really got this concentration in the 3C2A2 position here in terms of what people were being infected with. So we took those bloods, so we were still collecting those acute bloods. So now we took the acute bloods from that year and did microneutralization tests. The CDC ran some microneuts against a very kind of a panel of strains, but importantly, they looked at both the egg-derived Hong Kong virus and the cell-derived Hong Kong virus. And these are the levels of antibodies here. The two, the open triangle are the unvaccinated people, and the closed triangle are the vaccinated people. And so this is at hospital admission. What we saw are people that were, the biggest signal we saw were among people who were vaccinated, had a higher antibody titer against Hong Kong. However, that was limited to the antibody titer to the egg strain of Hong Kong, not the cell strain. So we see this huge disparity here between these two strains. And these two strains were specifically different based on a change in antigenic site B between the cell version of the Hong Kong strain and the egg version of Hong Kong strain. So now we see here the vaccinated people are kind of mounting a very robust, sometimes with my students, I say they mounted a very robust immune response to eggs, which is not it. It's mounting a very robust immune response to the egg-specific product, but is not necessarily matching with the cell-specific product here. So the other significant thing about the 2017-2018 year is that's the year that we started to see more uptake, not a lot of uptake, but more uptake in people that were beginning to receive cell-based vaccines. So what we did was, and we're not quite powered to do this, but we can still check out the point estimates to kind of look for patterns, that the egg-based vaccine, we looked at the Flucelvax, the cell-based vaccine, versus the egg-based vaccines for prevention against hospitalization, to see kind of in this kind of observational context, what are we seeing that do to the VE? And what we see when we look at the H3 and 2 estimates, that when you stratify and look only at Flucelvax, the point estimate is quite a bit higher than when you look at the estimate for the egg-based vaccine. Now, underpowered, we don't have we didn't have the numbers of egg-based vaccinees to be able to look at this specifically, but you do see a shift upwards here, and you don't see that shift at all in the B Yamagata down at the bottom. So that's sort of where we left off, 27 to 2018, and then we had some years of predominantly influenza B circulation. We had some additional years of mismatches of flu, but this is where we were at in our thinking about flu vaccine effectiveness. As of the 1920 season and that March 2020 season, where we suddenly cut off the flu season, and then we didn't see flu again for a year and a half. And so here is data from our community household cohort. This is a group of families with children that we follow every time they're sick, and we sample. Now we're going through a period where we sample them every week. They're very kind and generous people. This is the viruses that we were seeing starting. So here's March right here, 2020, and you can see when the K through 12 schools closed statewide, right where this dotted line is, and all the virus goes away. And they were still reporting illnesses during this time. We never stopped collecting swabs. We had staff in PPE standing in parking lots with a bucket, and they would drive up and drop something into the bucket at first until we got our collection system worked out. But as you can see, let's see, influenza is in this sort of peach color right here. But we have some influenza in the cohort prior to the closure, and we see some spotty detections throughout, but we really don't see influenza again in a real way until we get over here into later in this period. So October to October kind of 2021 into January 2022 right here is where we start seeing influenza come back in our community. And this reflects what people were seeing all over the country. So the 2020-2021 season right here is this pink line, and really no influenza to speak of. The next lower line above that is 2011-2012, which I like to brag is the first year I ever had an independently funded influenza research project, and had no flu to follow. So coming into the 2021-2022 season, when we start to see these triangles go up, we know we're going into the first flu season that we've had in a year and a half, because we didn't have anything to speak of in the 2020-21 season. So what did that look like? Well, this is what it looked like here in Ann Arbor. So we had a flu outbreak. It began in November and went into November, or began in October, went into November. And here, by the time New York Times got wind of it, we had 528 cases. We actually ended up with many more cases of influenza in this group of students. I also learned a lesson about the importance of telling university leadership that the CDC is coming to do an outbreak investigation before the New York Times finds out. That is a clue for the future that you can all take and learn from our mistakes. So the epi curve for the outbreak is here on the bottom, and you see it kind of starts in October, and the dark blue is the flu. Now, in terms of numbers, this is like in October and into November, we realized we were accumulating as many cases as in some years we based the entire national VE estimate on. So the thinking was, let's get out in front of this and let's calculate, we haven't had any US flu surveillance data to speak of in the last year. Let's get an early vaccine effectiveness estimate on this vaccine, and then we can really, start to act on that information. Our study population was, so the mean age, 21 years old. These are all people that were coming to the University Health Service. So this is for the most part people that are either living near campus or living in big houses on campus, living in the residence halls, and a handful of faculty members that still kind of attend the University Health Service for their care. And if you see the bottom here, you can really see the distribution. So most of these students were in off-campus housing, and then about 25 to 30% of students were in the official residence hall housing. And then we've got about 2.53% of students are officially in FSL, which is our fraternity sorority life housing. However, the houses themselves, fraternity sorority life houses in Michigan, only house a very small proportion of our whole fraternity sorority system. So there was a significant portion of these people living in the off-campus houses that were affiliated, kind of had the social interactions with the fraternity sorority system, even though they weren't living in the houses. And the vaccine effectiveness estimates, I was very excited when I heard that these estimates were ready, because I was very hopeful and optimistic that the vaccine was going to work that year, because we were in the middle of vaccination campaigns all over campus, and this was our estimate. We actually have our unadjusted estimates. We have exactly the same proportion of vaccinated over total for our cases and our controls. When you do the adjusted analysis, that when we restricted out people that had vaccine within 14 days, we restricted out people that had a positive SARS-CoV-2 test. We've done many, many, many sensitivity analyses, but the vaccine effectiveness was very null. We looked to see if there was any effect by where people lived, and the numbers were pretty similar and rather unimpressive in terms of whether they were living on campus or they were living either off campus or as part of fraternity and sorority life. So what happened? Well, what happened was that so we actually sequenced these right away, and this is Adam Loring's lab did this sequencing. His team did this sequencing as well. And what you can see here is here in the red dot in the bottom, that's A. Cambodium. That's the vaccine strain that we were currently delivering to students during the outbreak. And here, A. Darwin, this is the strain that was going to be in the Southern Hemisphere vaccine, but wasn't ready yet. And now you can see all of the infections that we have here. And so the blue dots, those were all infections that were on campus that we were getting from those swabs and mapping much closer to A. Darwin and pretty far away from A. Cambodia. Also using that sequence data, Adam's team was able to calculate the most recent common ancestor. And so we suspect the most recent common ancestor of this particular outbreak was about October 19th, which actually matches with, we interviewed a lot of the people in those first 2 weeks of cases, and it actually matches the interviews for where the most recent common ancestor probably was. And what it specifically matches in the interviews is fall break. This ad is specifically from a fall break trip that repeatedly came up in our interviews throughout a lot of the students, that they either had someone they were living with that went to this trip, or they went to this trip, or they knew somebody who went to this trip. And so the Just College Las Vegas fall break trip was likely is like $349, but the influenza is free on this trip here. We actually, our state epi actually released an epi alert about that trip as well, in terms of being a potential spread event. So remember I had last talked about the adaptations, to talk a little bit about what was going on with the virus, not just with the students. But when I talked about 2017, 2018, remember we were talking about a site B glycosylation change that likely was due to the egg adaptation and affected the vaccine effectiveness of the virus. We saw the same thing with this 2A2 virus as well. So that the 2A2 virus had actually had a loss of a glycosylation site in antigenic site B, creating again a mismatch between like an egg-driven mismatch between the circulating virus and the vaccine virus. And that actually, the circulating virus was then, because it being Darwin-like, was then updated the following year. So the following year, that Darwin strain became assigned the H3N2 component of the vaccine. So similar to what we had done in hospitalized adults, we collected serology in these students to look at, do microneutralization and look at what their serology looked like. So I just want to mention really quick, so we didn't have any incentives. We had little to no funding to carry out this work. And we just had a bunch of public health students and CDC EIS officers stationed at the health service asking people for their swabs and their blood. And 111 students who were acutely ill, meeting the definition for medically attended respiratory illness, agreed to give blood on the spot for blood draws. I mean, this was quite surprising to me. But what you can see here is actually we were seeing, both in the flu negatives and the flu positives, a fairly good antibody titer in the population against Cambodia, against egg Cambodia. So this is the vaccine strain on the left. If you look, though, at the cell Cambodia, much lower. So again, we see a lower vaccine titer against the cell-derived strains compared to the egg-derived strains. So sort of a repeat of those results that we saw from 2017, 2018 in the hospitalized adults. And if you go further to the right, and you look at Darwin, this is the virus they actually needed the antibodies to be against, and which would be the Darwin cell strain, and we see very low titers here against the circulating, the cell version of the circulating virus. When we break this data and then we stratify by flu A negative on the top, flu A positive on the bottom, and then you see the vaccinated and the unvaccinated next to each other. We see similar patterns that we saw in the earlier data. So you've got higher Cambodia egg titers in the vaccinated people in both groups. And then you see some increase in the egg Cambodia titers, but then very low across the board in the Darwin titers. It was interesting here, it was kind of validating to see the Hong Kong, so this is the prior vaccines that these students may have received, and we see some titer against Hong Kong in the vaccinated people, and unclear if this was due to prior vaccine history or cross-reactivity. And this probably goes without saying after you saw the vaccine effectiveness data, but we replicated some of those graphs that we did in the hospital where you look to see how the odds of infection changes as the titer grows on the log scale on the x-axis here. And you can see there was really no association between the odds of infection and the titer increase. We see very little movement. If anything, this Darwin curve is the one that shows the most here, but there was so little antibody there in the first place that this is sort of based on very little data as you get to those higher titers. So something I wanted to highlight about this is that, you know, the timeline of this outbreak investigation and by November 2nd, so there was 13 cases the week before flu. There were 47 cases on November 1st and 2nd. And between November 2nd and 10th, you know, we have a close relationship with the University Health Service. We got alerted, Michigan Department of Health and Human Services got alerted, and then CDC got alerted very quickly that we were having a very substantial and fast increase in flu activity. By November 14th, we already had EIS officers here on campus collecting specimens. Well, not collecting specimens yet, collecting specimens as of the 16th, we had the regulatory approvals in place. So this was MTAs, DUAs, and IRB approvals across the state and CDC and the university by the 16th. By the 18th, we were collecting bloods, and we were doing serial swabs. All samples and the enrollees, their data was linked real time with the state vaccination registry data. And by the nighttime of November 18th, we had our first VE estimate. And then by the 10th MMWR, the CDC Health Advisory, so the sequencing was also completed before the 24th. The CDC Health Advisory was able to go out within 2 weeks from notification of the outbreak. And the advisory included both vaccination data, and it included the sequencing data was confirmed by that point as well. So this was super rapid. This was a huge really fast turnaround time to be able to get an early sense of what the vaccine effectiveness was. And it's because we had this established infrastructure that's slowly been building from even since before the pandemic, where we've got our academic lab in partnership with the CDC, the MDHHS, and the university. The university, all of these partners also in partners with the county health department. And then Loring Lab has additional relationships with CDC and MDHHS and with us. And with those relationships came all of the regulatory pathways that you need to be able to do something quickly were already executed and in place. But the big challenge is, you know, so this on the left, the big challenge is what do we do with this faster surveillance? I think, you know, more and more of these systems were built as part of the pandemic, and there's a lot of effort and attention to trying to keep these systems alive. But the way that the flu world works is that we use that information in order to change the vaccine for the next year. So here, this is again data from our community household cohort, and you see from 2021 to 2022, that's the same year the sample size when you've got low VE and Hive is kind of challenged on a single year. You really have to do multiple year assessments, but I'm just going to show you the single year so that you can see. The vaccine effectiveness here was essentially null in 2021, 2022. So overlapping that time period where we saw the university outbreak. But then you look at 2022 and 2023 and you see an improvement. The H3N2 vaccine effectiveness estimate the point estimate is 48%. So that's for the Darwin strain in the vaccine. And that's kind of returned back to what we are used to seeing with VE. But it took a year to do so because of the vaccine development pipeline. It was a real public health communication challenge to have these results coming out in November and December, which is our prime influenza vaccine campaign time period. So the big challenge, the second big challenge I wanted to highlight kind of going forward, and I have just a little bit of very preliminary stuff to show on this, but this is a major focus of our work going forward, is, with this rapid surveillance and vaccine effectiveness also means that we're getting a lot more data about our changing virus and vaccine landscape in real time. And so now I think this is the third year maybe that we've been told to prepare for the triple-demic. And this is a perfect example of as these viruses are interacting and changing on the ground, we are tasked with the responsibility to try to understand how these vaccines are working real time on the ground. But these aren't really independent factors, these vaccines. So we've both got an explosion of viruses that people are now aware of. They were always around, but now I asked people if they've heard of RSV, and they have heard of RSV, so that's exciting. And we also have a lot of, we now have changes in the COVID vaccine, and we have the RSV vaccine for 16 and older. In our most rapid, I've shown you a mix of studies from longitudinal cohort studies and from VE studies, but the most rapid way to get the sample size that you need to be able to make decisions is to use the test negative design. And so this is essentially analogous to a case control design where you're enrolling people with a common case definition. Cough in an ambulatory setting in the hospital, or your case definitions will look like some sort of acute respiratory illness causing hospitalization. And then you're assuming, but in order, you test them to see whether or not they have the virus of interest, and then you ask to find their vaccination status, and you can use that to back estimate the vaccine effectiveness. But this design, even though it's the fastest and the most resource efficient design, it comes with some important assumptions, including your tested patients have to have that same healthcare seeking behavior, whether they test positive or they test negative. We also have to assume that vaccine does not impact your testing. And then that vaccine does not affect the symptoms in the people with a test positive infection, so that all cases, regardless of virus, vaccination, are more or less syndromically similar and would syndromically bring somebody to the doctor at the same level of severity. If you are a DAG person, here are some DAGs in the bottom that represents some of these ideas. And so here's what's currently happening under that paradigm. With influenza, this is working well enough. We've had several kind of studies triangulating influenza VE estimates with other study designs and other strategies and coming up pretty similarly. If the influenza vaccine is working and has a positive vaccine effectiveness, we would expect to have fewer people that are vaccinated and test positive. And we would expect to have more people who are vaccinated and test negative, and more people who are not vaccinated than test positive. And who are these people that are the people that are showing up at the doctors or at the hospital with acute respiratory infection and testing negative for influenza? And we've looked. So this is in 2014, 15, 20, and then 2015 and 2016. We actually tested everybody that was in our site of the Haven study against a huge panel of viruses to kind of talk about what else was out there. What are we comparing to as part of the test negative design? And we see a lot of RSV, a lot of rhinovirus, human metapneumovirus, other seasonal, non-SARS coronaviruses in these graphs. And when you take those and you do comparisons to say, what is the vaccine effectiveness if you are doing the standard test negative design, which is the first of each of these two mini forest plots. So influenza versus everybody else, all test negative. Here are our VE estimates. But then if you use just the other virus positive group as your control group, it's tough on the sample size to use that control group, but it was pretty similar. And then if you use a pan virus negative group, again, it was pretty similar in terms of who we were comparing to. Now, this made us feel pretty confident that we were a bit robust to the control group selection. But this was working so long as influenza vaccine was the only preventative vaccine out there. Now we've got COVID-19 vaccine, which prevents essentially the same syndrome that influenza vaccine presents. And so when you've got this general case definition, now you're picking up influenza, all those other vaccines and those charts I just showed you, but then also picking up COVID-19 vaccine. And the problem is that people who get the influenza vaccine and people who get the COVID-19 vaccine are often the same people. We have preliminary data that's showing that if you get a COVID-19 vaccine, it is highly, highly predictive that you'll have also gotten an influenza vaccine. It's actually, interestingly, in the IV network, where we've looked at this, we so far have not seen it traveling in the other direction. If you get the influenza vaccine, you're somewhat more likely to get the COVID-19 vaccine, but not to the same degree. So these two vaccines are correlated. They're working together. But then on top of that, if the COVID-19 vaccine, for instance, is working, that COVID-19 vaccine is actually going to remove individuals out of this group, this test negative group, for your influenza vaccine evaluation here. And so these two questions are becoming now intertwined in a way that's very difficult to untangle. There are now efforts to try to use differential control groups, take the COVID-19 out of the influenza control group and vice versa. Other ways to use sensitivity analyses to try to see this effect that we're working on methodologically to be able to correct for the other vaccine when you are doing an influenza estimate or a COVID-specific estimate. But wait, there's more. Now we have the RSV vaccine. We have the RSV vaccine, and then these are in 60 and older, who are typically some of our highest vaccinators, are the people that are 60 and older are now getting RSV vaccines as well. So now we've got this three-way interaction between people that may be getting the flu vaccine, the COVID vaccine, and the RSV vaccine, and how are we going to use the test negative design, which is still our most efficient and rapid way to understand how vaccines are working. How is that going to play out this season? So I am going to leave you with this thorny question that we don't have an answer for yet, but we are continuing to work on going forward. So hopefully I've demonstrated today that influenza epidemiology is a mix of, we're seeing repeats of old challenges like egg adaptation and vaccine match, but then new challenges as well. I think we're seeing examples of how one size or one VE measure may not fit all. And although all this rapid surveillance and availability of data is exciting, our production and approval pathways for vaccines don't move at the same speed. So what are we going to start to do with that tension? And then I think it's time for a lot more methodologic research on the test negative design. I know we're doing some, there's many others around, many others in Seattle that are doing this work as well. It's urgent because without it, we're going to lose our way to do real-time decision-making about vaccines going forward. And with that, I'm going to thank the many, many people that have worked on the various projects that I've showed here, and close there for questions.
Thanks so much, Emily. That was great and such an incredible overview of the work that you've done in Michigan over the last couple of years. I've only really read the New York Times articles and the MMWR, so it's nice to hear the story behind that. It looks like there's one question in the chat from Christian. I can go ahead and read it if that's and then have you answer it. So it's interesting to know if there's variation in predicting flu vaccination based on COVID vaccination stratified by type of COVID vaccine, mRNA, protein, et cetera.
So we have not gone down that road to look at that yet, but we do have. It's something that we're looking at, and I actually, one of the things that we're looking at is the other way. Also, is there changes in what flu product people get depending on their COVID vaccination history or the other flu product. What we're seeing with the flu vaccine data, sorry, I'm kind of pivoting to another element of this though. What we're seeing with the flu product data is it's so geographical. What product you get has so much to do with where you live because it's what was bought and provided in your area. I think in big cities, not so much, but in the more rural areas, which is much of Michigan, there may be only kind of one system that's buying and providing, and they may only be doing that with one product. So there's a lot of other factors that are going into that decision that may also essentially kind of overlay on socioeconomic, social vulnerability index, because it really has to do with where you live and what your access to care is.
Great. And there's a question from Lauren. Has there been enough uptake of FluMist to measure VE in this latest season? I guess maybe just globally, can you comment on FluMist?
Yeah. I mean, not in Michigan. I know the Michigan data really well. We've been tracking FluMist uptake over the last few years, and there's just been not enough to measure, at least in terms of our view of domestically, there's not been enough to measure.
You know of any national data? I don't think there is, at least from Seattle either. So
no, I suspect in the US market, it's so little that we're not going to get a US estimate, and we're going to have to depend on other countries for that one.
Great. We have a couple of questions for Trevor in the chat as well. Perhaps Trevor, would you mind just commenting on, I think there's a pretty, there's a theme about South Africa. And whether or not that is, does that provide a niche for certain variants to emerge? Does this have to do with potentially HIV and tuberculosis or immunosuppression in general in that population? And then just any information about JN1 being first isolated and the individuals in whom it was isolated.
So I think we still have this theme going on that is pretty unique, at least to my knowledge with SARS-CoV-2, where we have chronic infections that someone will be infected for a year's time, say that cat and mouse game within that one immune system really pushes the virus to evolve. And then we have the emergence of things like Omicron and JN1 and BJ1 and so forth, that really kind of push the virus along, and those emerge, and that is helping to capacitate overall global evolution of SARS-CoV-2. There has been a theme of Southern Africa, which we can maybe think that there's HIV prevalence plays a role in kind of harming immunity. So you get chronic infections. This obviously happened with Omicron and with JN1, but we also have other regions of the world, like BJ1 definitely seemed to emerge from India. And so it's not just Southern Africa. And however, I don't, to my knowledge, but this may exist, I'm not aware of like direct studies showing that HIV infection promotes these sorts of chronic infections. I'll stop there. Thank you.
Great. I think we have made our way through all the questions. So thank you so much to both Emily and Trevor for speaking to us today. And I hope everybody has a nice break. So.